everybody. Today we'll be discussing about some important concepts which will definitely come in the exam, that is isoelectric point Pi, pH as well as pKa. So let's start with isoelectric point Pi. So Pi is the pH at which a particular molecule carries no electric charge. So if you look at this zwitterion, where the ammonia molecule is having plus charge and the carboxylic group is having minus charge. So plus and minus cancel each other out and hence it is having no net charge. So when pH is equal to Pi, there is no net charge and the charge will be 0 of this molecule. Next, when we talk about pH and pK, it's often very confusing. What's pH, what's pKa, what's Pi? So let me just clarify that confusion first. So pH and pKa are simply convenient notions for proton concentration. So if pKa is high, it means that the hydrogen concentration, that is the proton concentration, is high in the solution and the solution contains amino acids. So if we talk at Pi at whole, Pi is given by a simple formula, that is pK1 plus pK2 divided by 2. So Pi is considered for the peptide as whole or amino acid if there is an amino acid in whole, and pKa is considered for each functional group. So we have ammonia as one functional group and carboxylic acid is another functional group. Generally you would consider carboxylic group as pK1 or pKa1 and amino group as pK2 and or pKa2. Yeah. So over here we see that the pKa1 value is 2.34. So 2.34 and the pK2 value is 9.69. Therefore 9.69 divided by 2. So this will approximately be 7.185. So this we consider as the physiological pH. That means the pH where the amino acid exists in plus and minus and neutralizes each other, and therefore the total charge on the amino acid is 0. So Pi is the charge on the amino acid, or if we are having a long chain of peptide like D, Y, K, L, if there is a long chain of peptide, then whatever is the charge on the whole peptide could be Pi, or it could be the charge on the amino acid, and Pi is given by pK1 plus pK2 divided by 2. So this is the first condition. Now we'll look into the second condition. So we have some other group called pKr, and pKr refers to the charge on the individual amino acid. So if we look at some amino acids like basic amino acids or acidic amino acids, consider amino acids such as aspartic acid or glutamic acid, or also lysine, and then we have arginine is R, and histidine. So all these are having some functional groups. These are having, because these are acidic, they are having COO minus functional group, and the basic ones are having NH3 functional group. So these are having some functional group. So even there should be the pKa value for even the functional groups, and hence there is pKa values for these functional groups also. Now if in a question, suppose if in a question they gave that there are pK1 is equal to 2.34, pK2 value is equal to 9.69, and they give pKr value as, suppose they give it as 3.4, then how will you find the Pi? There's a catch over here. I'll give you a simple catch. First thing, if there is any question like this which is giving you 3 P values, that is pK1, pK2 and pKr, just take whichever pK values are close to each other. Here 2.34 and 3.4 are more closer than 2.34 to 9.6 or 3 to 9. Hence we will calculate as 2.34 plus 3.4 divided by 2. The same formula applies. pK1 plus pK2 divided by 2. So here also we apply the same formula. Whatever is the answer will be the Pi. value for that particular amino acid, and this amino acid is nothing but aspartic acid. So there are particular pKr values for each amino acid, and there are also questions pertaining to this value. I will discuss everything. So let's discuss about this cation. We have already discussed about this cation, anion and zwitterion, but I'd like to reiterate once again. So here it's ammonium group that is having plus charge, and this is carboxylic group. It is having a proton. This is having an extra proton and this is having an extra proton. So whenever you are starting a sum related to peptide, finding charge on the peptide, remember that you have to always protonate the amino acid first. So the charge on this is +1 because this carboxylic acid does not give any charge. It's the charge of the ammonium. So the charge when it is fully in the cationic form, or when it is hydrated, or also called as protonated, it is +1. And also remember that whenever it is in the zwitterionic form, any amino acid, pH is equal to Pi. So the charge will be 0. And next we have the anionic form. So over here COO minus has already lost its charge. We only have NH2. So this NH3 has lost its proton and became NH2. So there is no charge over here, but COO minus carries a charge, that is negative. So anion has -1 charge. So keeping this in mind, we'll move on to the next slide. So now we'll talk about the titration curve of alanine. And this titration curve of alanine will clear off all the doubts we have on why there is pK1 value, pK2 value, Pi value, and how is it differing from each other. So let us take the first condition when the pH is very low. So let us consider the pH is 0.35. And at this pH, approximately 99% of the alanine molecules are positively charged. Let me draw this is the structure of alanine. If you see, there is NH3+, there's a positive charge over here. And this carboxylic acid is also protonated. So at low pH, everything is protonated, ammonium group and also the carboxylic group. So it is completely protonated. Now, at the first stage of titration, let us consider that we have 1 mole of alanine. And what do you remember if we say titration? Titration refers to gradual addition or removal of protons. If you remember in chemistry, we always used to titrate an acid with a base. So now we are having acid, that is COOH NH3+, because they are hydrated. Whatever is capable of giving a proton is called as an acid. Okay, let me write it here. Acid is a proton donor and base is a proton acceptor. Let me write proton acceptor. So now to the we already have amino acid, which is an acid. So to this we are going to add OH minus ions. This is considered as base. So we are titrating an acid with a base. Now what happens is when we supply exactly 0.5 moles of pK1, 0.5 moles of OH minus ions, we see that the carboxylic acid, whichever is there, it will start losing protons, and exactly half of the protons are lost. So this will come in a condition where it is half protonated, and we get an equimolar mixture of proton acceptor as well as proton donor. Acceptor plus donor. So it will be in this condition, COOH, COO minus. So they will be in equilibrium, and the rest of the structure remains the same. As you know, the rest of the structure is CH3, NH3 plus, and here also we have a H here. Sorry, because it's alanine, it will be CH3. Then we have C, we have NH3 plus, we have CH3. Okay, this is supposed to be H, not CH3, because we already have written CH3 here, and H. So we exactly have half the protonated form and half the deprotonated form, and whatever is between this and this range this is called as the buffering range. So we see that exactly when we add 0.5 moles of OH minus ions, there is 2 at the pH 2.34, there is exactly half the amount of protonated and deprotonated forms. And for alanine, the pH at the midpoint over here, pH is 2.34, and thus the COOH group has the pKa value of 2.34. Now let us consider the second situation when we are trying to increase the pH. Now titration corresponds to removing the NH3 group at this point when we have added 1.5 moles. Actually, it so happens that when we add 1 mole of OH ions to the amino acid, completely the COO turns into COO minus. In order to have an equal situation of COOH and COO minus, we just add 0.5 moles of the OH. Now, in the similar situation, if we add 2 moles of the amino acid, the NH2 group is completely deprotonated. But if we add just 1.5 moles, we see that there is an equilibrium between the same NH3 plus and NH2, the protonated form and the deprotonated form. And hence pK2 is also having a buffering range. Now whatever value this pK2 corresponds to is 9.69, and hence this is the pK value of the second group pK2, that is NH3 plus, and the range is 9.69. So at this Pi value, we see that when pH is equal to Pi, there is a zwitterionic form which is formed. That is, carboxylic group has lost its proton and NH3 group has not lost its proton. Hence there is one positive charge, one negative charge, which cancels out each other, and hence we get a zwitterionic form or a dipole, or a dipole which is carrying zero charge because it has cancelled out each other. So this is referred to as the physiological pH. And this is referred to as the isoelectric point or the isoelectric pH, or it is also a midway between, two pKa values. What are those two pKa values? pK1 plus pK2. And we know that whenever we are finding something in between pK1 and pK2, we divide it by 2, and hence arise the formula of Pi. So until now we have discussed about the titration curve of alanine, and alanine is having a CH3 group in the R position, that is in the functional group position. Hence it is not polar, this is non-polar. So for all non-polar amino acid, the titration curve is called as the diprotate titration curve. That is because only two protons donors are present. One is the carboxylic group and the other one is the amino group. But if we look at some charged amino acid such as aspartic acid or even the glutamic acid, we see that these both are carrying carboxylic group, extra carboxylic group. Apart from this one, in the R position there is a carboxylic group. So if you remember the structures of amino acid, you can recollect from the same. So because they are having some R functional group which is having another carboxylic acid extra, they are having one more proton. Apart from COOH, NH3 plus, they are having another COOH. So therefore this will come under triprotate curve. So now we'll study about these amino acids, and these are some of the pKr values. pK values of the functional groups you need to remember in order to solve certain questions. So first let's look at the basic amino acids. Okay, these are the basic amino acids which are carrying the NH3 plus group. And then we look at some acidic amino acids. These two are acidic amino acids which are carrying the carboxylic acid group. Then these two are the, even though they are polar, they do not have any charge on R. No charge on R. R is the functional group. So now let's go in detail. So aspartic acid and glutamic acid are acidic and they have carboxylic acid group. So aspartic acid symbol is D, and after D comes E, hence glutamic acid is E. Then lysine is K, arginine as the phonetic sound R, histidine is H, cysteine is C, and tyrosine is again the phonetic sound Y, tyrosine. So now we have to remember this symbol, so DEKRHCY, and also that what group they carry, either carboxylic group, either amino group, or what, if they are having any charge or not. We also have to memorize the pKr values. Don't worry, it will be very easy. Now aspartic acid is 3.7. So if you approximately remember the values, that's more than enough. 4.3, and lysine is 10.5. Arginine is having slightly higher, actually more pH, a more pK value. Then you have histidine, which is 6. So hence most of the enzymes are having histidine in their active site, because histidine's pKr value is 6. Then we have cysteine, which is having a OH group, and also tyrosine, which is having an OH group. Now let us look at this formula. These are for your quick recap. So let's consider some values. Let's draw a pH chart. So you have pH 1, 2, 3, so on until 6, 7, and then so on till 13 and 14. So if this is your pH chart, and if we have the pK1 value, if you remember, the carboxylic acid is having somewhere up between 2.34, and the NH3 plus group is having somewhere at 9.67. So if the pH is, so first condition, okay, let me change the color. First condition is, suppose if the pH is 4, first condition. So as the pH is 4 and the Pi of that carboxylic acid is 2.34, the pH is greater than Pi. As pH is greater than Pi, the carboxylic acid will lose the proton, it will become deprotonated. It will exist in the protonated form. So whenever we are solving a sum, we have to always remember that we put the protons. As the pH is greater, matlab pH has already crossed the Pi value, therefore proton is lost. As it did not cross the 9.67 value in the first case, that is in the pH 4, this will remain hydrated. So what is the charge on the amino acid? Here it will be COO minus, and here it will be NH3 plus. It will remain intact. So it will just cancel out each other, plus and minus. So the charge is 0. Now we'll take the second condition. In the second condition, where the pH is 10. So in the pH 10. Now if the pH is greater than as it is greater than 2.34, this will again become deprotonated. It will lose a proton. And as 10 is over here, as the pH has already crossed the Pi of ammonia also, this will also become deprotonated. That means it will just lose the proton and there is no charge on it. So now the value on this is -1 and the value on this is 0. So -1 plus 0 is -1. So at pH 10, the value of this amino acid is -1. That is, charge is -1. Now we'll take the third condition, where the pH is 1. When the pH is 1, the pH is still at the starting point over here. So when the pH is still over here, carboxylic acid will be in the protonated form and the NH2 will also be in the protonated form, NH3 plus. So it will have a +1 charge, and this will have a no charge because carboxylic acid H is there, there is no charge. So 0 plus 1, that will give you +1 charge. So that is how you have to keep in mind whether pH is greater or Pi is greater, in that way. So I have talked about all the 3 examples. Now let's solve an interesting question. So let's read the question first. What is the net charge of the peptide tyrosine, valine, arginine at pH 5? So they are asking at pH 5 what is the charge. The pKa's of alpha amino acid and carboxylic groups are, so pKa values is, just a second. So what is the charge on the amino acid? And the pKa of alpha amino and carboxylic groups are, so first they have given amino, so pKa or pK1 or pK2, anything, pKa1 or pKa2 or just pK1 or pK2. So pKa2 value of the amino group is 9.6 and alpha carboxylic acid is 2.3. Sometimes it will be given in the question, the pK values, but sometimes they won't give. So it's better to memorize. Apart from these 7 values, also memorize the pK1 value is 2.3 and pK2 value is 9.6. So these are all what you have to remember. All the pKr values, the symbols for them, only the 7 symbols, the pK values, and also the pK1 and pK2. Okay, and then the pKs of tyrosine and arginine side chains are okay, let me write this first. Tyrosine, valine, arginine. So first thing what you have to remember is, always we have from N terminal to C terminal any peptide. So over here it will be the N terminal and this will be the C terminal. Now because this is the N terminal, this is NH3 plus. Always remember, write in the protonated form. And the tyrosine symbol is Y, which is having a OH group. Valine has no charge. So firstly cancel out whichever is having no charge. Valine is a hydrophobic amino acid which does not carry any charge. Just cancel it out. And then arginine. Arginine is R. R is having 12.5. And arginine, if you remember, it's a basic amino acid which is having NH3 plus group. And also arginine is present in the C terminal. Therefore, C terminal carries carboxylic group. This is also in the protonated form. Write everything in the protonated form first. And the second thing, so whenever you're solving such kind of sums, you have to remember some things in mind. First thing is, always write it from N terminal to C terminal. So first we'll write it from N terminal to C terminal. In the question also it will be given in the same way. And second thing, consider the N group and the C group. The first one and the last one. First N and last C. So after you have written them, then you have to write down what is the functional group or the R group. Just make a note of it. And after that, fourth step is also mention the pKr values of these. Mention the pKr values. And that's it. You'll be able to solve the sum. So let's do the same thing. So we have written in N to C form. Then we have considered the first and the last functional groups, and then we have written the R, that is functional groups, and then we will write the pKr values. What is tyrosine value? Sometimes it's given in the question. So tyrosine is 10.46. Okay, this is 10.46. And what is arginine? 12.48. So what is the pH they asked? At pH 5, I want to find the charge. So at pH 5, it means ammonia group is having a pK value of 9.6. So over here the pH has not surpassed the Pi. That means this is still in the protonated form. If it is in the protonated form, the charge is +1. Plus the tyrosine is having 10.46. That means pH 5 has also not surpassed this. That means this is having no charge because OH is neutral. We have already written it's no charge, means it's having no charge on it. So this is carrying 0 charge. Now if you look at arginine, it is 12.48. This is also greater than 5. That means this is also in the protonated form. This is also +1. And then we have carboxylic acid. Its pK value is 2.3. As 5 is greater than 2.3, it has already surpassed the Pi value. That means it has turned into deprotonated form. That means it has lost the hydrogen. So it will become COO minus. When it has become COO minus, it gives one negative charge. So plus minus 1. So what is the total charge? -1 plus 1 cancels out each other. 0 has no charge. Therefore, the present charge on this particular sequence is +1. This is the answer.